Yes, class. Now let us talk about reproduction in bacteria. Reproduction in bacteria takes place by three methods. First is vegetative method, then asexual method, then sexual method. Vegetative method is usually done in favorable condition. Okay, when the condition is favorable, enough of food, oxygen, water, temperature is conducive, everything is there, then bacteria reproduces by vegetative method. And here the method that is followed is budding, where a small outgrowth is formed, and then the DNA material divides and goes into that outgrowth, and finally it separates from the mother body, and that is how it happens. So budding. Then the second is by fission, when the DNA material of the organism divides, and then there is a cytoplasmic division followed by cell wall formation. So that is fission. Bacteria E. coli reproduces by fission. So that's what. And in fragmentation, the body sometimes divides into two or more parts, and then all of them they make their own. They get their own DNA and cell membrane, cell wall, and they become individual in themselves. So this is how they reproduce by vegetative method. Now asexual method, it happens during unfavorable condition. When condition is little difficult for the organism, unfavorable condition, then asexual reproduction takes place. It takes place by spores. Spore in case of bacteria is usually endospore or cyst, though cyst is quite rare in bacteria. Then conidia. Conidia is a chain of spores in one branch. Say a branch that has come up, reproductive branch, and on the end of it there will be few spores which are known as conidia. So these are all asexual method in which special cells are produced for reproduction, and this happens in unfavorable condition. Now the third is sexual condition, which is actually reorganization of genes, or rather recombination of genes. Recombination of genes. It is actually recombination of genes, which is seen as sexual reproduction in bacteria. Now we shall discussing each of these three methods: conjugation, transformation, and transduction, in a little details. So this is reproduction in bacteria. Now let us go into the details. Now let us talk about sexual reproduction by conjugation. So here you will see two bacteria. One is having plasmid and fertility factor. This plasmid has fertility factor, and this is known as male or donor. The bacteria which has fertility factor in the plasmid, it is known as male bacteria or the donor bacteria. Then there is another bacteria which has got no plasmid and no fertility factor, and this is F minus. F minus because there is no fertility factor in it. This is also known as female or recipient bacteria. Now we have talked about pili. Bacteria sometimes have pili. So the male bacteria with this F plus factor, it has pili, and with the help of this pili it forms an association with the female bacteria, which is F minus bacteria. Now after this pili is formed, which is known as sex pili, there is a DNA replication in this plasmid of the donor bacteria. There will be DNA replication, and the extra DNA, this will go through the pili into the recipient bacterial cell, which did not have the plasmid gene. Now, the replication is completed and the plasmid has gone totally into the F minus bacteria, thereby making F minus bacteria F plus bacteria. Now this also becomes an F plus bacteria because it has got the fertility factor. The fertility factor is present in the plasmid and it has got the plasmid with the help of sex pili by F plus bacteria, and in turn it has changed into F plus bacteria. So the recipient has got F factor, fertility factor, and it has become F plus from F minus. This is what conjugation is all about. In conjugation, we have two bacterial cells, one is donor, one is recipient. Donor gives one F factor to the recipient and changes it into F plus bacteria. This is so there is a change in the bacterial cell in its genetic material. There is a change in the genetic material. So this is seen as reproduction in case of bacteria, and this method is known as it is happening by itself, just by the two bacteria. There is no third party here. So this is known as conjugation. This process of reproduction, this method is called reproduction, and this process is known as conjugation. Now this method of conjugation was first found by scientist Liederberg and Tatum in the year 1948 when they were doing some experiment on Escherichia coli. When they were doing experiment in Escherichia coli, a lot of experiment has been done on Escherichia coli, and lot of genetic properties, cellular properties we have come to know by this bacteria. So Liederberg and Tatum, they were doing some experiment on bacteria, and in 1948 they came across this method of sexual reproduction in bacteria. I hope you have understood conjugation method. Second method of sexual reproduction in bacteria, that is transformation. Here what happens, DNA, naked DNA from a cell, a bacterial cell is taken. This bacteria causes pneumonia, pneumonia in human being. That bacteria has been taken and it has been killed by giving heat. But the DNA material is still there, and some new bacteria is put into that culture where this bacteria is killed. The new bacteria which is put, it does not cause pneumonia, but it is put in the same beaker where this heat killed bacteria is, which causes pneumonia. Now after some time it is seen that this bacteria which is put after killing the earlier bacteria, this bacteria has become pneumonic. That means it can cause pneumonia now. Earlier it couldn't cause pneumonia, it is a non-pneumonic bacteria, but when it comes in vicinity of this DNA which is causing pneumonia, the DNA enters this cell and goes in the genome of the DNA of the bacteria and transforms the bacteria. It causes recombination. It causes recombination in this bacteria, thus changing it from non-pneumonic to pneumonic. This method is known as transformation, where non-infective bacteria becomes infective by coming just in contact with the DNA. Even the bacteria is not alive anymore. Bacteria is dead, but the DNA is still functional. And when this bacteria comes near it, DNA enters the cell, goes in the genome and changes it completely. This is known as recombination of genes, and the process is known as transformation of bacteria. And this process was discovered by scientist Griffith. Scientist Griffith in 1928, when he was trying to find out the cause of pneumonia, he came upon this method of sexual reproduction or recombination of genes in case of bacteria. So this is a process of sexual reproduction, and it is called transformation. So class, as we are discussing reproduction in bacteria, we have come to the third process, which is known as transduction. Transduction process was discovered by two scientists, Lederberg and Zender, in the year 1952. What happens in this process? Let us see. There is a bacterium, and this bacterium will be known as donor bacterium. And this bacteria will be infected by a phage virus, bacteriophage virus. Okay. So bacteriophage virus, what does it do? It will incorporate its DNA inside the bacterium, which will go and bind with the DNA of the bacterium and make a recombination of it. Okay. Now, as the process is, the bacteria will get destroyed and a lot of phage molecule will be created. This phage molecule will have phage particles. They will have recombined DNA because they will have a part of DNA of the donor bacterium. Now this virus will come out, this phage will come out, and it will infect another bacterial cell, which is known as recipient bacterium. Now what will happen? The recombined DNA which is there in the phage, it will come out in the bacterial cell and go incorporate in the DNA of this recipient bacteria, and thus bring about a recombination. So this is known as transduced bacteria, and this process is known as transduction, where a part of donor bacterial DNA is incorporated into recipient bacterium without any physical contact between the two. The transfer is brought about by a phage virus, So this is the process of transduction, and it's a process of sexual reproduction in bacteria, which mainly means recombination of the DNA.